So far, we've gone over the origin of the universe, what that looks like, and what the cause must have been like. Then we saw what this world actually looks like. That turns out it's actually really complex. It's literally geared towards life against all odds. Now we're going to investigate what the arrival of life and what life itself looks like. So the question is, how do we investigate the history of life? How do we know anything about the past? And the answer is simple. We look at clues left behind. In this case, we're going to take a look at the fossils. Now, I've always been intrigued by rocks, just the thought that they're a lot older than me. I like to think that rocks have a story to tell, especially in the case of the fossils. As biologist Louis Agassiz said, Darwin's picture of the history of life contradicted what the animal forms buried in the rocky strata of our Earth tell us of their own introduction and succession upon the surface of the globe. Let us therefore hear them. For after all, their testimony is that of the eyewitness and the actor in the scene. So let's hear the story that the fossil record has to tell us. Did life evolve or was it established? Now, if we're talking about evolution, I need to clarify some terms. The modern theory of evolution is the Neo-Darwinian theory. This is the synthesis between the classical Darwinian theory of common ancestry, along with our modern understanding of genetics. That's what the neo part means. So the Neo-Darwinian mechanism is natural selection acting upon random mutation. Natural selection states that the fit to survive are going to survive. They're going to thrive and pass on their genes. That's true by definition. That doesn't explain the arrival of new animals. That doesn't explain the arrival of new information, new body types. So we need to understand that, as Hugo de Vries said, natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest. What can explain the arrival of the fittest? What can get us this new stuff? That's what the random mutations part is about. They posit that mutations can bring us this new stuff, these new body plans, to get us these new creatures. So the question is, can mutations explain the arrival of the fittest? Can they explain where all this new stuff comes from? That's what we're going to investigate. So as we talk about evolution, just understand there are two important terms that are really distinct. There's macroevolution and there's microevolution. Macroevolution is large-scale change, usually having to do with the molecules to man view. It just means big change, macroevolution. Microevolution means small change, really small variations. Now, some believe that microevolution is actually evidence for macroevolution. The small changes with enough time actually lead to big changes. Sounds reasonable, right? Well, the fact is, if the Neo-Darwinian mechanism actually works, this is actually the only way it can happen. It must happen by adding up small variations, small changes over a long period of time, because extreme changes degenerate or become sterile. As Dr. Stephen C. Mayer explained, Darwin conceded that the beneficial variations responsible for permanent change in species are both rare and modest. Major variations in forms, which later evolutionary biologists would term macromutations, inevitably produce deformity and death. Only minor variations meet the test of viability and inheritability. So if the Neo-Darwinian mechanism does indeed work, it can only work by having lots of little changes. And that requires a long span of time. For example, if I tell you that, oh, you can't drive to school today, you got to walk. But then I tell you, oh, but you can only walk by taking an inch step each time. Now, since there's smaller steps, it's going to take you a longer amount of time. Well, the same with this. If we make them smaller steps, smaller changes, it's going to take a longer amount of time. So the question is there enough time? And does the Neo-Darwinian mechanism work at all? Now, the fossils are going to tell us a story. They're going to sing a song, and it's actually not too flattering to the view of Neo-Darwinism. I'm just going to give you 4 problems that the fossil record gives it. First, the sudden appearance of animal life forms. Second, the absence of transitional and intermediate fossils. Third, completely new forms and body plans. And fourth, major differences before minor differences. Don't worry, I'm not just leaving you there. We're going to unpack these. So first, the sudden appearance of animal life forms. Like I said, for the Neo-Darwinian mechanism to work, it needs to occur with small changes over a long span of time. Now, what we would not expect if that did indeed happen is the rapid arrival of complex life. We expect it to occur slowly and to start simple. But that's not what we find in the fossil record. Instead, we find a rapid arrival of different types of animals already complex. If we were to divide the history of the Earth into a 24-hour clock, in just 1 minute, we find 20 out of the roughly 26 animal phylae, that's major animal types, appearing abruptly in the known fossil record. In other words, just the fossils that we know of the first appearance of life, we already find 20 out of the 26 major animal types. That's incredible, how fast that is, and all these different types of animals that are already present. And they're very complex. So this already makes us start to doubt the Neo-Darwinian mechanism, as there's clearly not enough time, and there's already so much change present. That's just the first problem. Next, there's no ancestral precursors. There's no fossils of intermediate fossils or of the earliest ancestors themselves. Here on the left, there's a representation of a very simple tree of life. Now, as we can see, there are branches, which are the animals existing within kinds, but then they connect branches by saying, oh no, here's an ancestor that actually connected them both. They actually both came from this group of animal. And if you go further back enough, it actually connects at a trunk, the simplest life form. Now, here's the problem. In the fossil record, we find no intermediate animals. We find no ancestors. Instead, we find just the animals already existing within kinds. So if we take away what we don't have and just hold on to what we do have, just hold on to the evidence, we actually have a clear picture of life existing within kinds. Now, there are a few objections that the ones who really want to hold to a tree of life really point to. And they are, tectonic movement displaced the fossils into the sea, creatures were too small to be fossilized, creatures were not preserved due to soft bodies, and lastly, the sediment of the time wasn't ideal for preserving fossils. Now, let me just tell you, if you need to make excuses for why you don't find evidence for your theory, that already says itself enough in itself. But it's worse than that. There aren't just excuses, but these excuses aren't actually good. None of them hold. The fact is, we've been drilling in the sea for a while and still have not found any of these so-called displaced fossils. Second, we actually have plenty of small fossilized creatures, including fossilized algae. Third, actually 95% of these fossils that we find from the earliest life forms are soft bodies. So it's not like they're hard bodies and then we just can't hope to find what came before. In fact, even if they were all hard bodies and maybe the ancestors were soft bodied, you'd find some progression in there of creatures with some hard body parts, and yet we don't find anything. And lastly, turns out the sedimentary rocks prior to this were actually even better for fossil preservation. So basically, if there were animals before now, if there were ancestors existent, we would find them, even more so than what we'd find in this explosion of animal life in the fossil record. So if they were out there, we should find them. We haven't found them, therefore there's reason to doubt they're out there. So we can leave this tree of life and just hold on to the evidence we do have, pointing to animals existing within kinds. That's just the second problem. Next, we find new animal forms and new body plans. We find these vastly different creatures. The question is, to get these new body plans, what do you need? You need 4 things: new cell types, new proteins, new genetic information, and new epigenetic information. Now, for the sake of time, we're going to skip over the first one and go straight to proteins. Amino acids fold together to form proteins. So maybe think of this like how you do origami. You fold paper to get a specific structure. If you don't fold very well, you won't really get the structure. But it's even more complex than that with proteins. It's not just it has to look right. It has to be perfect because it has to fulfill a specific function. If it's not folded just right, it will not fulfill the function. The fact is that the average protein has 300 amino acids. The chances of them being folded just right is 20 to the 300th power. Now, to put in perspective, there are only 10 to the 80th power elemental particles in the known universe. So the probability of a protein fold being just the way it has to be for a specific function out of all possible amino acid sequences is just 1 in 100,000 trillion. In other words, you have no chance of getting a protein fold that is functional. Dr. Douglas Axe actually did a calculation of what he would take. What's the probability that you could get a new functional protein fold? So he calculated that if each creature to have ever lived, ever have walked on the earth, to have generated a new sequence, this is a very generous and even absurd assumption. So if each of these creatures would have tried one sequence, one new try of trying to get a new functional protein fold, only 1/10 trillionth of sequences would have been found. Now remember, just one in that vastly big number is a functional protein fold. And by doing this, we would have only tested out 1 in 10 trillionth of sequences. So we see that we have no chance of getting just one new functional protein fold. And that's just for one. The fact is we need whole new systems of proteins. There's not nearly enough time. And that's just proteins. Let's move on to genetic information. Now, just as in the English alphabet, we have 26 letters. In the genetic alphabet, we have just 4, A, G, C, and T, for adenine, guanine, cytosine, thymine. Now, just as in English, we can arrange letters into words and sentences. So in the genetic alphabet, you can rearrange it to get a specific meaning, a specific function. And this is really the point of it. It holds information. Now, genetic information is special. It's complex and specified. These two terms are really important. So definitely make sure that you're listening attentively to this part. Now, let's start with complex information. What does that mean? I'm going to introduce you to information theory, to Shannon information. This basically states that the more uncertainty removed, the more information is present. So for example, let's say I leave my house and I say, I'll be back later. There's not much information there, is there? No, there's still a ton of uncertainty left. Or let's say that I say, okay, I'm going out. I'm going to the diner across the street with some friends. Then I'm going to the movies. Then I'm going to go to my best friend's house, play video games, and I'll be back about 11 p.m. There's a lot more information in there because a lot more uncertainty is removed. That's what we mean here by removing uncertainty, that it can hold information. This is exactly what DNA has. It has complex information as opposed to simple information. See, simple would be like ABC, 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 ABC. Now you might ask, hold on, doesn't that remove more uncertainty because it's repetitive, it's predictable? Just seeing that small sequence, we can understand everything, every part about it. And I can see where you're coming from, but just think about this. Let's say that back to the story of me leaving my house. What happens if I say, I'm going out, I'll be back in a bit. I'm going out, I'll be back in a bit. I'm going out, I'll be back in a bit. I'm going out, I'll be back in a bit. Just because it's repetitive, it's predictable, doesn't mean it holds more information. This actually keeps it from holding more information. It's redundant and it even gets annoying. This is simple. The DNA is actually complex in that it's not repetitive, it's not redundant. It has the capability to hold information. Now, what do I mean by the capability? Let's imagine that I tell you, okay, you're gonna write a letter to your pen pal. And here's how you're gonna do it. You can write A, B, C, and then repeat. A, B, C, and then repeat. A, B, C, and then repeat. And so on so until the end of the letter. Now, that doesn't have information carrying capacity, does it? No, it can't hold meaning. It can't hold information because it's just repeating the same 3 letters. Well, that's exactly what I mean here by complex information. Complex information can hold information, has the capability to hold meaning, just simply because it's not repetitive. You can get lots of different combinations. For example, here, MHG, TV, VMB, GH, and so on. Is the capability to hold information. Doesn't mean it does, but it's able to because it's an improbable sequence. It's actually for this reason that the information in DNA cannot be the result of natural law. It cannot be the result of physical and chemical necessity, because that would produce rigid patterns, redundancy, not complex information. Because DNA has complex information, it cannot be the result of necessity. Now we know that the only 2 leftover explanations are chance or design. And that's how we get to our next one. DNA has specified information. Now, what is specified information? It's information that not only has the capability to hold information, but actually does. It holds meaning. So just check out this unspecified statement. It's the same one I included before of complex information, MHG, TV, M, GH, and so on. Now, because it's complex, it's the capability to hold information, it has the capability to hold meaning, but it doesn't actually hold meaning, does it? No, it's just gibberish. That's what we'd expect to result from chance. In fact, that was just me randomly typing on my keyboard. Specified information is where a specific function matters. As you can see here on my example of specified information is actually the book title of the book I'm writing. That'll come out in a few months, A World Like His. The specific arrangement of characters matters because it results in a meaning. It results in a function. And that's what specific sequence is. The specific sequence matters because it results in a meaning, it results in a function. It's the same with DNA. The fact is, when we take complex information, specified information together, that's what we know is the result of a mind. This is what helps us eliminate chance is the other one. For example, let's say that you walk to your house after being abroad and you find this banner that says, "Welcome home, Frank." Now, maybe you're like me that your name isn't Frank. But either way, you know that this is the product of mind. First, because it's an improbable sequence, is the capability to carry information, to carry meaning. And second, because the specific arrangement of characters actually does hold meaning. The specific sequence matters. And taking those two things together helps us cue in on the fact that this is the result of a mind. Now, the next question is, who's Frank? Did they replace me? But the fact is, it remains that we know this is design. And that's exactly what we find in DNA. There's complex information and there's specified information. Is the carrying capacity to hold information, to hold meaning, and then it actually does indeed hold meaning because it results in a function, a specific function, which depends upon a specific sequence. Now the question is, could you get this by randomly rearranging things? Or even if we assume that we did get it somehow, by chance or by necessity, could you get new information? Could you get additional genetic information by mutation? The answer is no, as we'll see in just a minute. Now, mutation has a few tools. Maybe it's insertions, maybe it's deletions, maybe it's just switching things around. For the sake of simplicity, I just used one tool, switching things around randomly. Now, if you take a specified sequence and start changing things randomly, you're invariably going to lose meaning. Here's my book title again. And I started randomly switching around letters. You can see that just after 4 changes, there's no meaning left, is there? No, everything is messed up. And that's just it. When you take a specified sequence, a specified group of characters, and you start randomly changing things, it's going to invariably lose meaning. It's going to lose function. And it's not just with replacing pieces. It's about adding in random pieces. It's about taking out random pieces. If you do something random to something that's specific, you're going to lose information. So the point is, mutating information invariably results in loss of meaning, loss of function. In the world of life, this is going to result in death. See, natural selection is actually going to narrow down on this and is going to end up killing the creature. Remember, the fit to survive are actually going to be the ones who pass on their genes. If something loses function, it's not as fit to survive. It's not going to pass on its genes. So this is exactly why natural selection is actually not a friend here. Any changes are going to result in the death of the creature. And so changes, mutations, are not the friend either. So we see that getting new genetic information is actually a big no-no. If you try to change things by mutation, you're actually going to lose information. So you find that life actually exists within boundaries. Next, epigenetic information. Now, epigenetic information, it's not what it sounds like. It's not additional genetic information. Epi means beyond. So this is information beyond the genes. For example, cytoskeletal arrays and membrane patterns. These include microtubules, centrosome, membrane targets, ion channels, electromagnetic fields, and the sugar code. Now you're like, okay, that's super sciencey. What does it do? What's epigenetic information? Well, let's imagine that I'm putting together a circuit board because I want to build a computer. I have all the pieces. Now, is that all I need? Are the pieces going to tell me how to put the circuit board together? No, I need information beyond the pieces to know how to put it together. I need a manual. Only then can I put the pieces together. Or let's imagine that I'm building a house. I get all the materials. I get pavement. I get bricks. I get tile. Is that all I need? No, I need information on how to put it together. I need a blueprint. I need an architect. And that's exactly what epigenetic information does. It's information outside, beyond the pieces that helps them come together. It arranges parts into higher level structures. So let's just assume, for the sake of the argument, that you could get new genetic information, which we already saw you can't, but let's just say you could. At best, mutating DNA produces new genetic information, but we need more than that. We need new epigenetic information, which is not found in DNA. Genetic mutations can't get us new body plans. So let's say I put my circuit board together, and then I get a new piece. I don't just need new piece, I need additional information as well as how to put that into what I already have. Or let's say that I put together my house, but I got new materials. I don't just need new materials, now I need an updated blueprint. I need the architect to help me understand where to put it in. That's exactly the point. We don't just need new genetic information, we need new epigenetic information. For every new piece, we need additional information beyond that piece to understand how to add it in. Now the question is, okay, so you need new epigenetic information. Could you get that? Could you mutate epigenetics? I'm going to tell you no for two reasons. First, the structures are actually too large to be vulnerable to the typical sources of mutation, such as radiation. And second, to the extent that cell structures can be changed, it's catastrophically harmful, resulting in detrimental consequences, death. So on one hand, we see that these things aren't vulnerable to mutation. It's very difficult to get to them. On the other, if you were able to mutate it, it would have detrimental effects, death. So once again, we see that life exists within boundaries. Change is not a friend. Change is going to invariably result in death. In fact, we just saw that even just one of these taken by themselves is improbable, if not impossible, to achieve. What's the probability of getting all of them? The fact is we need all of these parts and functions immediately, simultaneously. We saw that it's not believable to just get one of these pieces. What's the probability of getting all of them at the same time? You see, that's like putting together a motor. You can put it together piece by piece, but you can't have it running until it's finished. You can only run the motor once it's all put together. It's the same with life. The function isn't there unless all the parts are present. So we need new proteins, new genetics, new epigenetics, and new cell types in order for the new function, for the new creature to be able to function. If it's not able to function, it's going to die, and you're never going to get this new creature. So we need a huge change to go from creature to creature. And this is really the major point here. Major changes aren't viable, and viable changes aren't major. So we see that major changes are the only thing that can get us these new creatures, that can really take from one creature and make into a new creature. And yet these changes will never happen because it will kill the creature. And yet the changes that we do see, the changes that do happen, are not major. They will never get us these new body types. No matter how many changes you have that are small, no matter how much time, they will never get you these new body types. For example, to take a skeleton from inside the body to outside the body is not about lots of small little steps. No, you need a skeleton inside the body. If something happens, then that creature is going to lose fitness. It's going to die. The fact is, you need a huge jump. You need inside the body to directly outside the body. Otherwise, you lose function and the creature dies. So once again, the point here is major changes aren't viable. The types of changes we need will never happen because they kill the creature. And the changes that we do have will never get us the big changes that we need. This is exactly what Dr. Paul Nelson explained. If the only kind of mutations that can conceivably produce enough morphological change to alter whole body plans never causes beneficial and heritable changes, then it is difficult to see how mutation and selection could ever produce new body plans in the first place. And this leads us to the last point that the fossil record is going to give us. We find a top-down arrival of life. Now, according to Neo-Darwinism, we'd expect a bottom-up arrival, diversity before disparity. Now, to explain this, let's imagine the first life form is a hamster. Nobody claims it is. I want you to understand that, but this is just for the illustration. If the first life form is a hamster, we expect just one type of hamster, then over time, different types of hamsters, then over time, different types of rodents, then over time, different types of creatures, different types of animals. So you can see small changes before big changes, diversity before disparity. Instead, in the fossil record, we find just the opposite. We find a top-down arrival, disparity before diversity. In other words, we find all these major types of animals coming to existence at the beginning, and then we just find variations within those types. For example, we find the existence of fish, and then just different types of fish, but they're always fish. Or we find the existence of birds, and then just different types of birds, but they're always birds. That's exactly what we find in the fossil record, a top-down arrival. Different types of animals already coming into existence, not connected, and then just small variations within those types. So here are the 4 problems that we've investigated: the sudden appearance of animal life forms, the absence of transitional intermediate fossils, completely new forms and body plans, which genetics and epigenetics and molecular biology show can't happen, and major differences before minor differences. So ultimately, here's the death blow to Neo-Darwinism. Not only does the fossil record deny common ancestry, but our knowledge on molecular biology, genetics, and epigenetics reveal these large-scale morphological changes can't happen. In other words, we see due to the fossil record that these things didn't happen, that evolution didn't happen in the sense that there's common ancestry. And on the other hand, we see that through genetics, epigenetics, and molecular biology, these things can't happen in principle. So we see that they didn't happen and they can't happen. If Neo-Darwinism can recover from this, someone's pulling the strings, because it's down for good with those 2 objections. In fact, it goes far deeper than that. That's not the end of the problem. Theorists have calculated what the minimally complex life would be like. The minimally complex cell needs at least 250 proteins, each with an average of 150 amino acids. Now, what's the probability of producing that? The probability is just 1 chance in 10 to the 41,000th power. Now, to understand just how vastly improbable that is, that's 10 followed by 41,000 zeros. What does that look like? If all the events in the history of the universe were devoted to trying to create this one cell, the number of combinations tried would be only 1 out of a trillion, and that's just to have a 50% chance. So in other words, if everything that's ever occurred in the history of the universe, we're just trying to create this one cell, which is an absurd conclusion, by the way, then we would have only tried 1 out of a trillion opportunities to see different sequences, and that's just to have a 50% chance. Basically, there's no chance. And that's just for this one simplest cell to arise. Now, if that can't arise in the whole history of the universe, not even the history of the Earth, the history of the universe, then how can we expect the hippopotamus, the ostrich, the coral, the Venus flytrap, you and me, and the warthog to arise in just the short history of the Earth? We can't. Let's say that I tell you I can jump to the moon. And you tell me, okay, first go dunk that basket. Go dunk on that basket. And I go to do it, and I don't reach. If I don't reach to dunk on the basket, are you going to believe I can jump to the moon? Of course not. If I can't jump this far, I definitely can't jump up that far. Well, the same with evolution. If we can't get this little piece, this little simplest cell, then how can we expect all these complex life forms to arise? We can't. In fact, Richard Dawkins, the professor of zoology at Oxford, admitted that the message in the DNA of just one amoeba, one of the simplest life forms, is equivalent to 1,000 complete sets of Encyclopedia Britannica. 1,000 complete sets of Encyclopedia Britannica. Like, I don't know, I don't read encyclopedias for fun, but like, that is a lot. And remember, this isn't just random sequences of letters filling out pages in a book. This is specified information. Sequence matters. This is literally a parallel of our language. 1,000 complete sets of Encyclopedia Britannica. If you were just to pick up one book of the encyclopedia, you're going to assume that it's the result of a mind. Why is 1,000 complete sets of Encyclopedia Britannica any different? Or as Bill Gates, the founder of Microsoft, said, DNA is like a computer program, but far, far more advanced than any software ever created. If encyclopedias and computer programs need a designer, then what is to be said about DNA? What is to be said about life? See, everywhere else, forensic science, archaeology, computer design, we understand that design is a legitimate cause. Why is it any different when we get to the study of biology? Why? We find that the most epic and complex source of information And all the world is found within ourselves, within life. Why would we make a distinction there? So what have we seen? We've seen that life, even at its simplest, is complex. All the right parts need to be present in the right place, at the right time, and in the right sizes. In other words, life is irreducibly complex. Even at its simplest, it's complex. There's no such thing as a simple life form. The 3.2 billion character genome is not a random sequence. As I said, it's a specified sequence. It's a specific sequence, a message. Now, our repeated experience confirms that messages only come from minds. See, this isn't just because we saw that chance doesn't work, necessity doesn't work, therefore it must be design. It's not just that we narrowed it down. No, this is evidence for design. We know that when we've complex and specified information, that actually points to design. So in other words, this isn't just negative evidence against others. This is positive evidence for intelligent design. From our experience, which is empirically detectable, we know that messages only come from minds. And the fact is, we can't reduce a message to the medium, to the material. Like I said, I'm writing the book A World Like His. It's going to be on ink and paper, but the message is not reducible to ink and paper. I hope that if you read it, you come out with a better education than just like, ink and paper. No, the information transcends the ink and paper. It's the medium, it's not the message. As evolutionary biologist George C. Williams explained, you can speak of galaxies and particles of dust in the same terms because they both have mass and charge and length and width. You can't do that with information and matter. Information doesn't have mass or charge or length in millimeters. Likewise, matter doesn't have bytes. This dearth of shared descriptors makes matter and information two separate domains of existence, which have to be discussed separately in their own terms. So we see that information and matter aren't two separate domains. They're in two different areas. Whereas Dr. Philip E. Johnson explained, the content of the book or the computer program is not specified by the physical or chemical laws governing the medium. If it were, all books would be alike, or perhaps would differ according to the qualities of the ink and paper used to write them. In fact, the content of the message is independent of the physical makeup of the medium. The message comes from an author. Ink and paper are merely the media. Similarly, the information written in DNA is not the product of DNA. Where did the information come from? Who or what is the author? So to illustrate this point, imagine that your father is reading the daily newspaper. He's reading an article, and your brother is reading the same article on his computer. And lastly, your little sister decides to write that same article on the sand. Now, is the information the same? Yeah, it is. And yet, the material, the medium, is completely different. On one, ink and paper. On the other, metal and plastic. On the third, sand, crushed rocks, and shells. So we see that information transcends the material. The material might hold the information, but the information itself is not reducible to the material. If I walk up to the beach where your sister's writing, and I see the writing, I'm not going to say, oh, it came together by necessity or by chance. The waves came together and made the sequence. No. First, it's complex, so it's improbable, which means necessity can't do it. Second, it's specified as a specific sequence, so chance didn't do it. I rightly assumed it was designed by an intelligent being, your sister. And that's just it. We know that information comes from design. We saw that the genetic information can't have been changed. So it's been complex since the beginning. And we saw that there was indeed a beginning a couple weeks ago. So in other words, there was nothing. Then this designer wrote this message. So the question is, if the genetic code isn't reducible to its medium, to the DNA, that is indeed a message, who wrote it? Dr. Frank Turk and Dr. Norman Geisler said, when we conclude that intelligence created the first cell, or the human brain, it's not simply because we lack evidence of a natural explanation. It's also because we have positive, empirically detectable evidence for an intelligent cause. A message is empirically detectable. So once again, this isn't just negative evidence against other theories. This is positive evidence for intelligent design. A message is empirically detectable. We can detect it. Or as Dr. Michael Beakey said, life on Earth at its most fundamental level, in its most critical components, is the product of intelligent activity. The conclusion of intelligent design flows naturally from the data itself, not from sacred books or sectarian beliefs. I haven't quoted a single Bible verse or any other holy book in this presentation. This is science. And we see that design flows naturally as we follow the evidence where it leads. Whereas molecular biologist Dr. Douglas Axe said, the reason we perceive purpose in inventions is precisely the reason they can't occur by accident. They exhibit an organized functional coherence that can only come from deliberate intelligent action. They are conceived from the top down and constructed from the bottom up. They may operate by nothing more than physical causes, but they certainly don't originate that way. Or as Dr. Stephen C. Mayer said, standard materialistic evolutionary theories have failed to identify an adequate mechanism for cause for precisely those attributes of living forms that we know from experience only intelligence, conscious rational activity, is capable of producing. So we've made a pretty strong case for intelligent design. And so what do we do with this? Well, you might find this very colorful illustration by Dr. William Dempsky very helpful. He said, if a creature looks like a dog, smells like a dog, barks like a dog, feels like a dog, and pants like a dog, the burden of evidence lies with the person insisting the creature isn't a dog. Now, he's not talking about dogs here. That's an analogy. We've seen that there's clear evidence for design, for mind behind life. Here's just a recap of what we looked at. We saw the explosive arrival of major types with no ancestral precursors. We saw the impossibility in the history of the universe to produce the simplest cell. We saw the incapability of the Neo-Darwinian mechanism to produce new body plans. We saw that the message in life points to a mind, to a designer. This is strong evidence. This is a strong case for intelligent design. The burden of evidence lies with the person who says life was not designed. That's what Dempsky is talking about. So we've seen that there is indeed intentionality. The whole universe was created with the purpose of this planet being able to hold life, our life. So if there's purpose to this life, to our life, is there a right way to live? That's what we're going to talk about next week. If there's a purpose to our lives, then there is a right way to live. And that's going to close our case for the existence of God. We saw the origin of the universe, the complexity of the universe, the origin and complexity of life. Now we're going to see the purpose to life. Having seen that, there's so much more we could cover, but we have a very strong case for the existence of God.